Sometimes a single assay or sample experiment fails and should be removed from downstream analysis to allow more sensitive and precise detection of real differences. These failed experiments can be detected and easily removed from Array Studio data objects. For example, we can run a principal component analysis, or PCA, which attempts to detect the components that contribute to the greatest variance in the data. Different samples, such as tissue types or treatment versus control, should be separated. Outlier samples can also be revealed. Choose your input data, which variables and observations to use, and name an output. You can specify either a 2 or 3 component analysis. If you specify more, multiple plots of 2 component analyses will be shown. Let's make a 3 component analysis. Let's specify a group. If you perform a 2 component analysis and check this box, the plot will show ellipses indicating the confidence interval for each group specified here. With a 3 component analysis, a 3D plot will be shown. We can see that 2 samples stick out from the rest of the data. We can choose to exclude these data from further analysis. Choose the selection arrow, click one, hold down Control, select the other. In the details window, we can see that this is breast control sample 6 and breast treatment sample 3. Now click Exclude Selection. Under List, you will find a list of the samples that were not excluded, and a new PCA will be run on the samples that were not excluded. Our list of selected samples can be used to create a new omic data object. Click Omic Data, Subset, choose our source object. We will use all variables, but only include some of the observations. Our new object still contains 34 variables, but only 22 of 24 samples. Now let's perform hierarchical clustering on our data. Click Microarray, Pattern, Hierarchical Clustering. We can choose our new data object or filter based on our list. We can choose which kinds of trees to calculate, whether or not to normalize, and what kind of dendrograms to generate. For this example, we won't normalize the heat map. However, we will increase the scale. Right-click and choose Change Color. We can add a color bar to easily see which sample group each of these samples belongs to. Let's add source tissue and group. Our data cleanly separate by source tissue, but not so much by group. If we filtered out the genes that were fairly consistent between tissues, we might see a better separation. We can create a list of variables to include by selecting some of the data, holding Control between selections, right-clicking List, choose Add List from selected rows. In this case, the subsetting didn't help because there are still too few genes that were actually affected by the treatment.